factors are appear in the organisms. So now we are going towards the gene therapy. Gene is nothing but the segment of DNA, and how that gene it is used in the diseases, various diseases. So the treatment of diseases, there are the various diseases, and they are treated with the help of that gene, and that's why it is called as the gene therapy. That means here, by replacing, altering, and supplementing the gene, the disease is cured, the disease is treated. This is called as the gene therapy. There are the different types of the diseases. The diseases, many of the diseases, they are depend upon the gene. For example, here the cystic fibrosis, hemophilia, sickle cell anemia, thalassemia. These are the different diseases. They are related to the gene. That is, the gene is responsible for diseases. But the another diseases like a high blood pressure as well as the cholesterol, such diseases, they are depend upon the gene as well as the environment, So there are the two types of the diseases. Out of that, some diseases, they are depend upon the gene as well as some diseases, they are depend upon the environment as well as the DNA. So when we apply such a DNA, or the gene for the treatment of the DNA, it is called the gene therapy, and this gene therapy it is used in many ways. That is, the gene is delivered in a replacing or the missing or the defective gene. That is, the gene is replaced for the missing and the defective gene. That is, whenever there is a defect in that gene, we can replace it, we can deliver the gene for that defective gene. As well as here we can deliver the gene to destruct the cancerous cells. You know that the gene, cancer is depend upon the oncogene. So we can replace or we can deliver the gene to destruct such oncogene. As well as here the cancerous cells revert back into the normal cell. So whenever the cancerous cell it is produced, it convert into the normal cells by delivering such a gene. Then to produce the vaccine against the viral and the bacterial gene. So the bacterial gene and the viral genes, they are used to produce the vaccines. To generate the immune response. You know that whenever the pathogen it enter into the human being, it produces the diseases, and so to generate the immune response, we can transfer the gene into the human being. As well as for the antigen expression, what is the meaning of that antigen? Antigen means here on the pathogens, the particles present on the surface of that pathogen is called the antigen. So for that expression of that antigen, here we can deliver the gene. As well as to impair the viral replication, the viral it get replicated, and to impair that, here the gene can be delivered. And whenever there is a injury, to damage, there are the damaged tissue. So to heal that damaged tissue, we can deliver the gene. So on that many ways, we can deliver the gene. As well as there are the different types of the delivering the genes. The first is the ex vivo delivery, as well as in vivo, and as well as here the use of virosomes and the bionic chips can be used for the treatment of gene therapy or diseases. Here the ex vivo condition means when the gene is transferred into the cell. Here the gene is, suppose this is the cell, and in that cell when the gene is transferred into that cell, that means here the cell, you know that all organisms, they are made up of the cell, and when such a cell it is isolated from that human being, and then in that cell when we transferred the gene, then it is called the ex vivo condition. That is when cell it is out of that body, then it is called the ex vivo condition. In such cases, here the viral and the non-viral vectors are used. In case of the Parkinson's diseases and the neurological diseases, here the ex vivo condition is used. In vivo condition, here the cell it is transferred into the human being in a targeted organ, and targeted especially in a cancerous cells. Here in vivo condition or the in vivo therapy is used. That is, in vivo means here directly the gene is transferred directly into the or the deliver into the human being targeted organ, and ex vivo means here the cell, it is isolated from that patient and then the gene is introduced, then the gene is delivered, then it is called the ex vivo condition. In case of that Parkinson and the neurological diseases, the ex vivo condition or the ex vivo therapy is used. And in case of the cancerous tumor, here the in vivo condition is used or the in vivo therapy is used here. The virosomes are also used for the gene therapy. Virosomes are nothing but the liposomes or the lipids plus inactivated HIV virus. This is the HIV virus, inactivated HIV virus and the lipid. So the lipids plus inactivated HIV virus is called the liposome. Such virosomes are also used for the gene therapy. Then here the bionic chips are also used for the gene therapy. It is nothing but it contains a single living cell and embedded in a tiny silica particle or the tiny silica circuits. The cell acts as a diode or the electrical gate, and such bionic chips are used for the gene therapy. Then there are the different forms of the gene therapy: germline gene therapy and the somatic gene therapy. In germline gene therapy or somatic gene therapy, what happens here in somatic gene therapy, here the especially the organ, the gene is transferred in organ. That's why somatic particle or the somatic organ, it is nothing but the lungs, muscles, liver. In such cases, the gene is delivered, and this is called the somatic gene therapy. In lungs, here the gene is delivered. In muscle, here the gene is delivered. In liver, here the gene is delivered. In vivo condition, so this is called the somatic gene therapy. And germline gene, such somatic gene therapy, it is used in the cancerous, rheumatoid arthritis, blood disorders, as well as the sickle cell anemia, muscular dystrophy. In such cases, here the somatic gene therapy is used. In germline gene therapy, what happens? The germline cells, they are present in the ovomes as well as the sperms and the embryo. In such cases, here the gene is transferred in sperms, here as well as in ovum, here the gene is delivered. As well as here in the ovomes, here the gene is delivered. And in embryo, the gene is delivered, and this is called the germline gene therapy. You know that the germline cells, that from single cell, from entire organism is produced, then it is called the germline gene therapy, and it having a capacity to divide and redivide. That's why it is called the germline gene therapy. And it is the chromosomes, they are transferred, or the such gene, the delivered gene, it can be transferred into the next generation, but it is not applicable in the human being because of the technical and the ethical issues. It is not applicable. So as like this, here we have completed the gene